Today, we're diving into some absolutely groundbreaking research that could one day completely change how we think about treating some of the most devastating neurological conditions out there. This is a story about cracking a code and unlocking the brain's own hidden power to repair itself. So let's start with a really fundamental and frankly terrifying question. What happens when the brain sends a command, like move your hand, but that message never arrives? For people with conditions like ALS or a severe spinal cord injury, this is their reality. The communication line between the brain and the muscles has been cut. It's this broken connection that's at the very heart of the problem scientists have been trying to solve for decades. All right, to really understand the incredible solution these researchers came up with, we first have to zoom in and see the problem at a microscopic level. We have to look at the brain's broken wires. Okay, so think of your nervous system as this incredibly complex electrical grid. Well, the main power lines, the ones that carry commands for voluntary movement all the way from the brain's cortex down the spinal cord, those are a very special type of cell called corticospinal neurons, or CSNs for short. You can think of them as the absolute superhighways for movement. Now, in ALS, these vital neurons start to break down and die. And in a spinal cord injury, their super long connections get physically snapped. Either way, the result is the same. That superhighway gets shut down. All the traffic coming from the brain just grinds to a complete halt. And for a long time, this was a massive roadblock for researchers. I mean, this quote from the paper we're looking at says it all. Scientists just didn't have a good way to create and study these specific CSN cells in a lab dish. And you have to ask, how can you possibly fix something if you can't even study it properly? But this is where the story gets really interesting. The researchers started to think, what if we don't have to build these neurons from scratch? What if the solution is already there, hiding in plain sight inside the brain? And that's the big question that really drove this whole project. What if, scattered throughout the adult brain, there are cells that have this hidden ability to become new neurons? You know, like a kind of biological backup system, just sitting there, waiting for the right signal to get to work. And incredibly, they found them. These sleeper cells, and don't worry about the technical name, SOX6 plus NG2 plus progenitors, are a type of support cell that's just scattered all over the brain's cortex. And the really important part, they aren't rare. They are already right there, perfectly positioned where they might be needed for repairs. But of course, there's a catch. Even though these cells have all the genetic potential to become neurons, they're normally locked into being a totally different kind of cell, a non-neuronal cell. It's like they have the blueprint for building a neuron, but it's locked away in a safe and they don't have the combination. So the researchers went looking for that lock, and they found it. It's a protein called SOX6. Its whole job is to act like a biological brake, constantly pushing down and suppressing the cell's hidden neuron-making program. So to reprogram these cells, the team knew they had to find a way to take that brake off. But here's the thing, the team didn't just want to release the brake. They wanted to grab the steering wheel and drive that cell to a very specific destination. And this is where they basically cracked the cellular code. To pull this off, they engineered what you can only describe as a genetic master key. It's this little package of multiple genes they called NVOF. And this key wasn't just designed to unlock the cell's potential, it was designed to give it a complete step-by-step instruction manual for how to become a cortical spinal neuron. So how does this master key actually work? Well, it's pretty brilliant. It does 3 things all at once. First, it flips the on switch for the really critical genes that tell a cell, hey, you're a neuron now. Second, at the exact same time, it flips the off switch for any other career paths, telling it, nope, you're not going to become one of these other cell types. And third, it gives it very specific directions to make sure it becomes the exact type of neuron they wanted. It's incredibly precise. So they had the sleeper cells, they had the master key. The only question left was, would it actually work? Well, the results were nothing short of a stunning transformation. I mean, just look at this. On the left, you've got the progenitor cells in their normal, simple state. And on the right, after they were given that NVOF master key, boom. They've changed into these complex, branching cells that look almost identical to the real corticospinal neurons they were trying to create. And this wasn't just a cosmetic change, not by a long shot. These new cells were seriously high-fidelity copies. They had the right shape, the right molecular ID tags, and, most importantly, they were electrically active. They could actually fire signals just like real working neurons. They weren't just looking the part, they were acting the part. Now, to really get why this is such a huge deal, you've got to see it next to the old ways of doing this. Before, you'd get these kind of generic, confused neurons. They had the wrong shape, and they still acted a bit like their old selves. But this new NVOF method, it produces neurons with incredible precision with a totally clean, new identity. It is a massive leap forward in control and accuracy. So, what's the bottom line here? What does this all mean? Well, this isn't just some cool experiment that works in a lab. This research is building a foundation that could literally rewrite the future for people with neurological diseases and injuries. The impact is happening in stages. Right now, today, scientists can use this to create way better disease-in-a-dish models to figure out exactly what's going wrong in ALS. In the near future, that means we can test potential drugs much more accurately. And the long-term dream? Well, this work lays the essential groundwork for one day using the brain's own cells to repair itself. You know, the researchers themselves really say it best in their paper. This work sets a precedent and a foundation. It's the starting point, the launching pad from which all this future research can take off, aiming for that ultimate goal of actually repairing broken circuits in the brain. And that leaves us with this incredibly powerful and hopeful question. Now look, we are still a long way from this being a therapy used in people, but this research unlocks a profound new possibility, that the key to rebuilding a damaged nervous system might not be something we invent, but something we simply unlock, tapping into the incredible potential that's already hidden inside our own cells.